This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 759, recorded on May 21, 2021. Yeah, how about that? I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. I'm lagging here because I got to get up the weather. Says 82 on my screen, and my window says it's cloudy. It's not unpleasant. There's some more rain in the forecast, but promises. We need more.
Right here, it's 25 and cloudy in New York City. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it's 25 Celsius, that is, here, 77 Fahrenheit. Humidity is 41%, it's clear skies, and it's gorgeous, and we've also had a really dry time, but it's nice.
And also from here at Columbia, New York City, Amy Rosenfeld, welcome back.
Thank you. I'm unclear. Maybe your window's really dirty, but I see blue skies out my window.
It is blue. You're right. It is blue. It's
cloudy in your window.
It's tinted, and so, yeah, I see that.
You know what? You need cataract surgery. That's what.
Yeah, it is. But it's not that deep blue sky. It's kind of a grayish blue sky, right? And they're kind of
grayish
little
blue. Again, what are you looking?
Come on down here and look out the window, okay?
I'm in the attic, so I can't just pick up and leave.
Maybe you just need a color blindness test.
No, the other day
I thought color blindness only did red.
Brilliant blue. All right, I got to get a second camera here so we
there
can
are multiple forms of color blindness.
Get a weather report.
I'm more familiar with those who can't do red. I think that was what Richard and Dubin couldn't do, was red.
Okay.
Not Kathy, Amy. Sorry.
Happens all the time.
Kathy. People object when you use first names and you don't identify people, because someone wrote, "Who's this person that Amy keeps naming?"
So our collaborator for Zika, Richard Valley and David Dubin, were colorblind, and you couldn't use red in some of the graphs.
We couldn't use red immunofluorescence either, right?
Right.
That would be a problem.
Yes.
Boy.
My buddy Nasseen's colorblind. He used to carry. I used to keep a pair of those. What are they? Red, green 3D glasses by my computer, because I don't understand this. But if he wore those, he could. Kind of put them on and off and that kind of stuff. He could differentiate some of the colors that I was pointing to.
Yeah, you can make, if you put a filter in front of your eye, you can make colors disappear and reappear.
Don't forget, Dixon will be with us shortly. He's a little delayed today, so he'll pop in any minute now. And I want to remind everyone about vaccine education town halls at asv.org/education. Go sign up for one of them, and you can get your vaccine questions answered by a couple of experts.
More important than ever, we're in the mopping up phase here. Those of you who are hesitant or who have questions, there's no reason that your questions should go unanswered.
All right, today for your listening pleasure, we have two brevias in a regular paper, and the two brevias are on coronaviruses, but not SARS-CoV-2. How about that?
There are other coronaviruses.
All right, one of them is a preprint, one of them has been published. The preprint medRxiv is entitled Emergence of Porcine Delta Coronavirus Pathogenic Infections Among Children in Haiti Through Independent Zoonoses and Convergent Evolution. And this is from a group of investigators at the University of Florida, Gainesville.
What do you know?
And none of them are in your old department, right? Which was micro and immuno.
Right. I didn't see any from that department, but of course, several of these people are familiar to me. The lead author is an acquaintance of mine. One of the middle guys who probably did the structural stuff. The lead author is John Lednicki. And there are a whole bunch of other authors. A middle author here, I'm sorry if I leave people out. David Ostroff is an acquaintance of mine. He probably did the structural work. The two others that I'm familiar with are Marco Salemi and Jay Glenn Morris, who is the director of the Emerging Pathogens Institute.
That's a relatively new institute, right?
Yes, it is. It
recently spilled over from another department.
It has an interesting birth story. As a matter of fact, it is Dick Moyer's brainchild. Okay. Though, to give credit where credit is due, though it has undergone a serious morphogenesis since Dick's original brainstorm. I would say it is... Actually, I can pretty much pinpoint it. I would say it's about 16 years old.
Okay. Now, Dick was the chair of your department, right, for a while?
Yes, but then he was kicked upstairs to be some sort of associate dean for research or something like that in the College of Medicine. I forget exactly the title, where he had responsibilities to advance research, the research enterprise in the College of Medicine. And this had been he had this idea for a long time to get together numerous different disciplines, like even climatology, meteorology, and that kind of stuff, and epidemiology and basic science and et cetera, biostatistics, what have you, to have a sort of a holistic view of emerging infections. Okay. And what is now the Emerging Pathogens Institute was the result of that vision. I think it's realized in a slightly different fashion, but it exists nevertheless as a result of his efforts.
So as you heard in the title, this is a Delta coronavirus. So that's one of the four genera, right? Coronaviridae family is divided into four genera: alpha coronavirus, beta coronavirus, Delta and gamma coronavirus. And, the coronaviruses that infect us humans are alpha and beta coronaviruses, as far as we know so far. So this is a unique idea here, that there is a Delta coronavirus. And porcine Delta coronavirus specifically is an interesting history. It was first reported in Hong Kong in 2012, and then 2 years later in the Americas. It infects pigs, particularly piglets in the GI tract, can cause a viremia as well. The virus reproduces in the epithelial cells, both the large intestine and the jejunum and ileum, and can cause dehydration as a consequence of diarrhea, which is not good in piglets. This study is out of Haiti. Well, the samples are from Haiti. And interestingly, I didn't know this, but in the 1980s, they had an issue with African swine fever in the pig population there. So they eliminated all the pigs, at least the domesticated pigs. I presume there are pigs running around in the forest, feral pigs that they didn't.
I would expect so.
And to get rid of this virus. And then they brought new pigs in from North America, from U.S. and Canada.
Sort of extreme imposed herd immunity.
Yes. Herd elimination.
And there's no, as the author said, there has been no reported porcine Delta coronavirus in pigs in Haiti. It's elsewhere. So that's the background that you need. And so what was done here, they had collected plasma samples from kids at two different schools in this case with febrile illness of unknown origin, right? Kids get fevers. They took blood samples, and this was between May 2014 and December 2015, 369 samples. And 3 of them turned out to have coronaviruses in these samples, or at least I think one had infectious virus, and the other, all 3 were PCR positive. And none of them were very sick. They had fever. I think one had abdominal pain and cough. They all recovered. They're all okay. So these were stored samples that, for one reason or another, they decided to look at. Kind of accidental, right? And I think we ought to be doing more of this on a more routine scale. But they extracted nucleic acids, and they could find. Well, what they did is they tried virus isolation. They looked for alpha and flaviviral RNAs by RT-PCR. They were negative. So they threw this onto Vero cells, a bit of the plasma. And one of the cell cultures developed cytopathic effects. They purified the nucleic acids, and they turned out to be positive for coronavirus RNA. They did an unbiased amplification and sequencing, and they could see coronavirus there. And they ended up getting 3 whole genome sequences from these 3 kids' samples, basically. And they are porcine. They're most similar to porcine delta coronavirus, but actually one of them, case 1 and 3, which were identified 4 months apart. So case 1 and 3 were 4 months apart. They're highly similar to a pig isolate from China, which was identified a year after one of these kids' samples was taken. And the other is the Delta coronavirus is most similar to a pig strain from Arkansas, identified in 2015. I mean, we're talking about 99.97% and 98.9% nucleotide identity. So 2 distinct, let's say, call them clades here,
right,
related to 2 different viruses in 2 different countries, which, who knows how they got there, right? So they think these are 2 separate zoonotic transmissions to these kids. When and how, who knows?
Now, these Delta coronaviruses, are these the ones they also infect birds? Is that right? Where did I see that?
I don't know.
They can.
Can they infect birds? Yeah.
Yeah, but I don't think that they go between birds and pigs.
Okay.
I don't think it's like flu, if that's what Alan is thinking about.
You just want to know if Delta viruses infect birds.
Yeah, so Delta coronaviruses.
Yeah, I think that's where that comes from.
Avian Delta.
There are avian coronaviruses in that family.
Right. Okay. Yeah. So this virus probably didn't come from a bird.
Right.
Probably came from a pig.
Yes. And
is most closely related to these 2 far-flung other pig Delta viruses.
Right. And it's not like, you normally think bird to pig to human.
Right.
Flu.
No.
So it's not like that.
I mean, they say that, because the pigs came to Haiti from the U.S., and we know that this virus is in the U.S. pigs, so maybe that's where it came from. But, as they know in their beginning, no one has found this virus in pigs in Haiti. So, or maybe it's there and we don't know about it, right?
How closely have we looked?
Probably not. And also, what are the kids doing interacting with pigs? Or maybe one of the parents worked in a pig farm?
So I gather the pig farming in Haiti is scattered all around the place, and a lot of it is subsistence.
That's right.
Pig farming, so it's like the family pig.
Got it.
And the kids would be quite likely taking care of the pig.
Okay. So I'm not talking about huge industrial pig farms here. Yeah. Okay.
No,
they may also have those, but I gather a lot of the pig farming in Haiti is happening just at home.
Got it.
I'm really taken by this. Is a bit of a sideshow, but I'm really taken by this figure of the cytopathic effect, and I want to riff on that for a second for the listeners. Down a technical detail here. One way to recognize the presence of a virus is to throw a sample onto a culture of cells and ask whether the cells show any manifestation of having been infected. And this can be a lot of different things. They can round up or, you're going to do it for us.
Is that, can you see that?
Yeah, that one. Yeah. They can round up or all different kinds of things. This is pretty subtle. It's a little distracting. It's a little distracting that the fields are different colors.
Right.
But if you look closely enough, the thing I notice is the vacularization.
Yeah. Right.
There are big vacuoles, but if you look even closely, you can see what they describe as granulation. Some of the cells have like more speckles in them than others. But if you just kind of sit back a minute and look at the two and try and equalize the color in your head, you can see that they're different.
Yeah, they are.
But
different.
it's subtle.
Boy, is it subtle.
Right?
Yeah.
Yeah.
Yeah, I think we would have picked that up, right?
Yeah, I would have picked that up. Oh, yeah.
All of us.
If you were looking for it.
I wouldn't have, but yeah, if I was looking for it, I wouldn't have believed myself. I would want to do it. Yeah. I would want to passage it and do it several times. Okay. And I would be dragging people in from the hall.
Don't tell them which is which. Yeah.
Don't tell them which is which.
Yeah,
exactly.
So the CPE was only from one sample, but they did get full genomes from all three. So that's, pretty good evidence that there was some virus in all three of them. And this is serum, right? So they're probably infected, but, it'd be...
Not all infections are lytic.
Yeah, I know.
Right.
I mean, I maybe want to... I don't know. What would... They cannot conclude that these kids were all infected with porcine Delta coronavirus, right? There's sequences there, but only one gave rise to infectious virus. Maybe,
only one gave rise to infectious lytic virus.
Well, these are all very
you should have done like an EM or something, right?
Yeah, that would have been good to see particles. Yeah. Yeah.
In particular, you cannot conclude that they were sick because of this.
That's right. That's correct.
They were sick and they found this.
Yes.
All right. I would imagine that down the road, now that coronaviruses are on the radar, we're going to see more of this kind of thing. And maybe over time, you'll find a strong association between some coronaviruses and illness. But it's hard to do the experiment. How the heck do you prove in humans that a virus causes the disease?
Unless it's causing an outbreak, you just have to regularly identify it in people who are ill. Right now, a note here, of the 369 samples, these were the only 3 that were positive. But apparently many of these kids had fevers, right? Because that's why they collected, right?
That's how they got the samples.
So other things.
It might be interesting to do serosurveys just in the population. I mean, you probably can't get a hold of these kids again, but if you could, see if they have antibodies, but also just go around to people who have pigs and see if they've got Delta coronavirus antibodies.
Yeah. The other part of this that's interesting is, so they do a time to most recent common ancestor. So you can look at the sequences and do molecular clock analyses and say, what's a recent common ancestor? And so one of them dates back to October 2014, and the other dates to 2011. So they think these have been circulating in pigs for a few years and maybe even in people. But they do have a signature. They compare the amino acid sequences with the porcine viruses, and there are 5 amino acid changes that they call a signature of these 2. The 3 isolates from the kids all share these 5 amino acid changes in ORF1AB and 2 in the spike, which they say maybe are things that got selected during reproduction in humans, right? Which we don't know because there's such a small number of data.
Or maybe they got selected in reproduction in pigs locally, and then
who knows?
Yeah.
But they are signatures that are not found in other
porcine
deltas. Yeah.
Hi, Dixon.
Hello, everybody.
Oh, it is Dixon Despommier. We were just talking about a porcine Delta coronavirus.
I heard you talking. That's why
Dixon,
I came on.
do you know anything about pig raising in Haiti?
No. Okay. I know they're almost like free-range pigs.
There you go.
They come and go.
Independent confirmation.
There we go.
But it says
they're peridomestic. They're called peridomestic, and they share their diseases and their food and their thoughts.
When I was a kid, I used to visit my father's family in Italy,
All right.
Starting in the 1960 when I was 8 years old. And his family lived in the middle of nowhere up in the mountains in Italy. And they had, they had grapevines and they had a pig every year.
You bet.
And this pig was kept in a little shack across the road from the house next to the grapevine. And when they cooked in the kitchen, they had a bucket and they would throw all the scraps in the bucket and then bring the scraps at the end of the day to the pig. And I would love to go and watch them. They would put the bucket in there and the thing would bury it would bury its face in this thing and eat it.
Oh, absolutely.
Oh, man. And I would sit there for hours watching this pig, it was lying there. And I would sometimes pick up pieces of plants and feed it to it. It would eat whatever I gave it.
So we ought to screen you for Delta coronavirus antibodies.
Right. And then at the end of the year, they'd take it out and prosciutto it.
Yes, and then the next time we went, there was prosciutto from that pig, but they had another pig then. They would get a new pig. Yeah.
When they slaughter the pigs, they save all those scraps and they feed them back to the pigs because they will eat meat as well.
And then
that's how Trichinella gets transmitted.
They also had chickens, and the chickens were just wandering around. Like you didn't know whose chickens were whose, right? Because all these houses. And
that's right.
I remember my grandfather, my father's father, he would periodically grab one of these chicken. And right there, and he had a little workshop below the house. He would just slit its throat and bleed it out, and then we'd have it for dinner.
Exactly.
So maybe I have avian influenza viruses too. Sorry about that.
That would explain a lot.
This brings back memories. I had forgotten about this. I took pictures of these pigs. I used to love. I'd never seen a pig before.
We are too far from our food.
Yes.
We are.
But it says in the conclusions that these pigs often are free roaming and not uncommon in rural and urban areas and constantly in contact.
Throughout South and Central America, the whole situation is like that.
This is another peripheral comment, but I'd be interested in knowing how long it was between the elimination of the existing pig population with African swine fever and the reintroduction of pigs, because I'm a little surprised that there was an African swine fever hanging around for some period of time that could come back. Okay.
And the odds of having gotten all the pigs seem kind of low.
I suppose it helps to be an island.
When they become feral pigs, then they're a real problem.
Sure.
Yeah.
By the way,
problem in this country.
So African swine fever is a pox virus, right, Rich?
Well, poxy.
Poxy.
Okay.
Epoxy.
I like that.
Epoxy.
It's in its own family.
Okay.
But it's a cousin.
Is it an asfarviridae?
Yes.
Okay.
It's its own family.
So
it's we used to describe it as a pox virus in iridovirus clothing. Okay.
Iridovirus capsid, please.
So I think these feral pigs are probably infected with porcine delta corona. We don't know it, and they infected the kids. But they say in the last paragraph, we do not find evidence that they call it human porcine delta coronavirus represents a major human health threat. But I think it just points out this was just picked up accidentally, right? So who knows how many other spillovers
It's amazing what you see when you look.
Right.
Hey.
This is the under the streetlight thing, right?
Yep.
Okay. And with coronavirus, with now 3 spillovers of coronaviruses into humans, the streetlight has gotten bigger.
It's true.
All right.
But it's not unusual, to be honest, that they can't pinpoint the virus to human disease. I mean, we're still arguing about EB68 and AFM, right?
We are?
Some people are.
I'm convinced.
Yeah, tons of people still are.
Reviewer number 3, right?
Exactly. and lately, there's 5 of them all in one battle. It's amazing.
Now it's a problem.
But yeah, I mean, if you talk to some people that we well respect at the CDC, he says to you, there's tens of thousands of things in that sample. And I'm not 100% clear that it's EB68.
I think the way you get convinced is if you have a virus that infects many, many thousands of people, and each of them are PCR positive for that virus, say a nasopharyngeal swab, and then you can be pretty sure that that virus is causing that disease, right?
So I wonder if the direction we're going ultimately is to have sort of metagenomic sequencing on, on clinical samples routinely. Okay.
Yes.
And that would inform not only what disease agents might be out there, but probably be the first line of defense against new spillovers.
Yep.
I've been pondering over this. How do we keep this from happening again? And it's a complex problem.
But if you do, let's say someone comes to the
doctor, metagenomics.
Yeah, you're going to find a lot of sequences, right?
You do real-time PCR like BioFire. That's what they do.
Well, if you're going to do that, then that's
directed.
Somewhat selective. Yeah. Okay. I'd like an unbiased approach. So we're not looking under the street lamp. We're looking at the whole landscape.
The problem is you're going to get a ton of stuff, right?
Yeah.
And you may not know,
right.
With the EV68 story, right? You sequence kids' snot and you get a lot of viruses and bacteria, right?
Yeah.
Right.
That's
I
right.
mean, and most of the time when you do metagenomics, like even for looking at the microbiome, it's still biased because you use 16S and you don't pick up things like chlamydia. So I could have the most rampant chlamydia infection whatsoever all over the place, and I will never test positive by 16S.
But I think people usually refer to the 16S analyses as 16S analyses, and metagenomics is the term. Most of the people in the field who I've talked to would apply metagenomics to mean a more unbiased, open-ended.
Yeah, but it began with when you talk about
with 16S.
Yeah, yes. When you talk about microbiome studies, they began.
Yeah, and that still dominates the field because that's a whole lot more specific.
Right, but
obviously you're not doing 16S if you're looking for viruses.
I do think
there's nothing that's equivalent, so you have to do things that are somewhat. You're going to have to do things that are somewhat biased.
Yeah,
you can't sit there doing random hexamers and then trying to do the bioinformatics for a week.
Amy, what else is chlamydia?
It is a bacterium.
Yeah, but is it 16S?
No, it doesn't respond.
That's right. So I asked you, which S does it have?
Oh, I'm not sure.
By the way, for those who are not following this, 16S refers to the RNA contained in the small subunit of a bacterial ribosome, and it varies in a defined fashion so that one can, by sequencing specifically this molecule, get a handle on what bacteria are in a sample.
And the S stands for Svedberg units.
Yes.
Yeah. Boy, that's
now you want to explain that one?
That's how far it settles in a centrifuge.
Who the heck was Svedberg?
But actually, Dixon, I take it back. I misspoke. It does have a 16S.
Okay. That's okay.
It's just the sequence of that RNA is so very different that it doesn't anneal with the comment. Yeah.
Right. What else falls into that category that you would be concerned about of the chlamydial-like 16S? Do you have other microbes that you're worried about catching as well that don't get detected by a metagenomic survey?
Well, there's a lot of urinary tract bacteria. So I did this when I used to study trying to correlate the urinary tract microbiome with incontinence in older ladies.
Wow.
Wouldn't it be nice to have, if it were cheap enough to get a metagenomics on everybody who came in, you could store it and then go back to it if there were a pattern,
I'm betting money on this. Okay.
Yeah,
before I die, I'm going to live to be 100, by the way.
Okay.
And before I die, so within the next 28 years, this will be a reality. And by the way, for those of you out there contemplating a career, there's a career in bioinformatics. Computational biology. Because somebody's got to sort all these data out.
There's a huge demand still for those folks.
I'll say.
Yeah.
There's some special
good
people.
ones are very hard to find.
They're very hard to find because they can make 10 times more money in industry.
That's true.
Well, I had one who was trained as a virologist. Actually, he got his PhD with Diane Griffin, then went to an immunology lab, then decided he was going to do bioinformatics. We had a collection of gram negatives that I believe were facultative or anaerobes. We did some shotgun sequencing. He came back to me and he said, gram positives. And I was like, no, gram negatives. He said gram positives. I said, no, gram negatives. He said, let's go to the microscope. And he told me my gram stain was broken.
You believed his bioinformatics, huh?
Yeah, I had to walk away at that point.
He drank the Kool-Aid.
But
Yeah,
those
at
folks
that
can
point, I had to walk away.
with the data analysis skills that they develop as bioinformatics folks, they can make a ton more money weaponizing people's data against them at Facebook. So,
Yeah, right. All right, so we have a second isolation of this is a canine coronavirus from a pneumonia patient in East Malaysia. Novel canine coronavirus isolated from a hospitalized pneumonia patient, East Malaysia. And this is clinical. What is the name of this journal? Damn.
Clinical infectious diseases.
Thank you.
CID. Yeah, clinical infectious
CID.
disease.
And this comes out of a variety of places. The Ohio State University, the University of Gondar, which is not in Lord of the Rings, although it sounds like it. That's
Gandalf.
that's in Ethiopia. Ohio State has an office in Ethiopia. Interesting. Duke University. Then we have a little bit of the Peking Union Medical College and a hospital in Malaysia. And a university, Segi University in Malaysia. And also Duke appears to have something in China going on there. And Singapore, Duke NUS Medical School. Wow, Duke is all over the place. Malaysia and Singapore. How about that? I gave a TWiV at the Duke NUS Medical School in Singapore at the end of 2019. That was a lot of fun. They listened to some of them listened to TWiV there. All right, so what do we have here? This is
Let's briefly do the authors. The first two are Vlasova and Diaz, who contributed equally, and then there's Damti, Zhu, Hokto, Lee. Linda Safe is a familiar name, and Gregory Gray, who, in fact, while I was in Florida, was affiliated with Emerging Pathogens Institute. So we have some professorial spillover here, I suppose.
So Linda Safe is also on the other paper. She's at Ohio State Vet School and works on pig viruses. And Steph Langle, who is on Immune, did her PhD with Linda. With Linda Safe. So this one is about canine coronaviruses. So these are alpha coronaviruses. They cause enteric disease in dogs. There's there are a bunch of them. There are two canine coronavirus 1 and 2. Number 2 has been we found it in 1971, and CCOV1 was discovered 3 decades later. And then we have some feline coronaviruses that are going to come into play here, as you'll see in a moment. And apparently, at least one of these feline coronavirus originated from canine coronavirus 1 through gene loss and recombination. So there's a complex history among these dog and cat coronaviruses. Anyway,
dogs and cats living together in the street.
Oh boy, how about that? Oh my, right? That's the last part of it. Yeah. All right, so these are patients with pneumonia. They had 301 nasopharyngeal swabs from hospitalized pneumonia patients. This is 2017 to 2018 at 2 hospitals in Sarawak, Malaysia. And 8 of these had been confirmed before by PCR to be positive for canine coronavirus, PCR and sequencing. So they all came from one hospital, Cebu Hospital. 7 of them were less than 5 years of age. 4 were infants. And they say most were from the people who live in rural or suburban longhouses or villages. So rural population in contact with wildlife, most likely. All of them recovered. So they extracted RNA, and as I said, 8 of these by PCR and sequencing were positive for canine coronavirus. 2 of the 8 patients. And so 8 of these samples were inoculated onto canine cells. One of them produced CPE, and they do have a figure. Have you looked at that, Rich? Do you like that CPE?
I have indeed. No, they have a picture of the virus, an EM of the virus. They don't have a picture of the CPE. Remember, folks, cytopathic effect, that's CPE. Now, they have a picture of the virus. I didn't take the picture.
Okay.
I don't want to cast aspersions. I've seen clearer pictures of coronaviruses, but we do have here round blobs on apparently, I guess this is negative stain with clear evidence of the corona, spike-like corona. So I'm buying it.
All right, so they were able to take the supernatant of these cells and infect fresh cells and made CPE again, so they were able to passage the virus. They extracted the RNA and they used for complete genome analysis. But all of these patients had a similar virus by sequencing. So this whole genome sequence is interesting. So it's highly similar to canine coronavirus, some specific strains. But what's interesting, and they call it canine coronavirus human pneumonia 2018 virus. They think it's a novel strain within the alpha coronaviruses. What I thought was interesting is that the spike actually appears to be a recombinant of the canine coronavirus and a feline coronavirus spike. And as you know, you've certainly heard that coronaviruses undergo recombination prolificately. Is that a word?
Profligately.
Profligately.
Or prolifically.
Prolifically. There you go.
Prolific.
And so this should be no surprise to you. So this strain is a novel.
Profligate, I think they spend their money freely.
Exactly.
Yeah.
Prolifically.
So it's a recombinant strain, and it is nothing like we've seen before in dogs or cats. So this is out of people, and maybe this can actually infect these people. Although, again, as we can't really prove that it caused any of their pneumonia.
No.
But these were nasopharyngeal samples, right?
Do you think dogs and cats, when we're not looking, commiserate and sit down and discuss the world and pass the time of day and ask how's the family, etc.? Because in the daytime they chase the hell out of each other.
If you go on YouTube, apparently dogs and cats are interacting extensively.
There are often, depending on the household setting or where they are, domestic dogs and domestic cats often get along just fine.
Yeah, that's true.
They sometimes have very cute interactions.
Yeah.
It's,
So I would guess in the wild they might interact to some extent, right?
Wow.
As I was reading this and the other paper, I was reminded of the sort of bird flu era. Okay. And how that spawned a number of experiments with serosurveys and et cetera of the human population to try and assess how frequently it was that various bird flu viruses encountered the human population, and maybe, a glancing blow, this kind of thing. Yeah. And it's, as I recall, the bottom line is it pretty frequent, that you can get, a transient, non-transmissible infection with an influenza virus that may or may not cause disease. But there are people running around that are seropositive to bird flus that are not readily transmitted in humans.
Yeah.
Okay.
Well, there was a paper that Vincent held on for later dates that talks about bat flus and how they are really adapted for bats. But there are periodic spillover events into humans, and they don't do anything. For a later date, it's in the to be discussed folder.
We have a number of overlapping arcs in the pandemic era here, and with at least in this country and places in Europe, the waning of the pandemic, it's appropriate to be talking about, okay, how do we keep this from happening again?
Yeah.
And these papers are relevant to that very question, because this is evidence that these viruses are testing the boundaries all the time. And it's not just coronaviruses. There's going to be a lot of other viruses doing the same thing. And so how do you deal with this? I don't have an answer to that, but it's an important question.
And it's not just human spillover. So here we're looking at a virus that may have gone back and forth between dogs and cats. And then We see it in these human sequences. And I've got to say, it's a lucky thing that nobody happens to have a virology institute located in Sarawak, Malaysia, because otherwise people would be screaming, "Oh my God, it came from the lab!" Right? I mean, this it's a recombinant dog-cat virus. How else could it possibly have happened?
Yes. So also in the folder of to be later discussed is another paper that talks about SARS-like viruses that were identified from horseshoe bats at the bottom of Russia that just came out.
Bottom meaning southern part.
Yeah.
Right.
Oh,
bottom part.
I don't know. Okay, I guess I wasn't clear.
We'll look at
the
that
southern
one, yeah.
region towards Malaysia.
The bottom of the United States would be Death Valley.
That's right.
Cool. More to come.
Okay.
So I think. I'm sorry. Go ahead.
Oh, go ahead.
No. Finish your thought. It's all right.
I forgot now.
Okay.
Sorry.
So a couple of points. First, these never went anywhere, obviously, these human infections, but it just shows that there are spillovers, and we need to look for them more than we do. These are both kind of, experiments of convenience, right? They didn't go looking.
Samples of convenience.
And the other is that, in the pre-show, which is something no one hears except us, we chat, and sometimes we do it more than others. Today it went on for 15 minutes. We're talking about, the origin of SARS-CoV-2, and someone pointed out that this is an example of spillovers happen all the time, man. You don't need a lab to make a spillover.
That was me.
Yeah.
Yeah. I think that spillovers happen far more common than we know.
Yeah.
And you only know when you have either enough people in a localized area that are dead, or, that's what you do, or you have very severe disease and it's uncommon. And then
you have somebody who happens to be doing some new technology in sequencing and notices.
Yeah, I noticed.
Right.
Yeah.
So these papers, you've got, a few years ago, these kids showed up with pneumonia. They got over their pneumonia, blah, blah, blah. Go back to those samples and somebody goes and sequences them and says, hey, a novel virus in there. So back to Rich's idea of at some point in the future, in Rich's lifetime, we're going to be just sequencing everybody, who has a, unknown disease and finding all these viruses. And I think when that starts happening, there's going to be a whole lot of, oh my gosh, it's this. Oh my gosh, it's that. Because we will have turned on the lights and we'll see all the roaches running. And now I've realized that this was going on the whole time.
I've gotten a bunch of emails from people who you can now send your poop to a company to sequence your poop virome, your fecal virome, sorry.
Right.
And these people write and they say, oh my gosh, the most common virus in my poop is plant pepper mild mottle virus. What should I do?
Eat fewer peppers.
And I said, don't worry about it. It's with pepper. You like peppers? Good. Eat them. It's not going to reproduce in you, but that's exactly what we're talking about. You find something, you don't know what to do with it.
Well, I think a lot of people are not aware that, we're just swimming. It's a sea of viruses out there. Okay.
It's a sea of viruses.
Everything living is just swarming with viruses, most of which don't do anything. So anytime you rub up against, your cat or your dog or any other creature, there's an opportunity you're bumping up against their viruses too.
Yep. You bet. A sea of viruses, indeed.
More like an ocean.
An ocean. Okay. By the way, these two papers in normal times would have been ignored by pretty much everyone. They're picked up everywhere. I think NPR had a story on it. Science Magazine had a commentary. I saw this morning The Scientist had a commentary. And I would say
Times, Nature.
Pre-pandemic, none of them would ever have covered any of these. But now they do, which is fine. It's good. All right, one more paper. This is in Cell Reports Medicine. I'm not sure we ever did a paper in Cell Reports Medicine. Why don't we make Cell Reports for every possible field you can think of so you can make more money?
Yeah.
That would be Cell Reports TWiV.
There we go.
Serotypic evolution of measles virus is constrained by multiple code-dominant B-cell epitopes on its surface glycoproteins. Thank you, Amy, for pointing this paper out to me. The authors...
It's something we're very interested in.
The authors are Munoz Alia, Nace, Zhang, and Stephen Russell from the Mayo Clinic in Rochester, Minnesota.
I wonder what Roberto thinks of it.
I don't know. Maybe he will let us know because he does listen at least when we discuss his papers, he comments on it.
Well, I've got to say, it's kind of refreshing to have a paper that's 4 authors, all from the same institution.
Yes.
Just like very straightforward. Here's our little group doing this.
Yeah. And by the way, so this is an interesting topic because we've talked about it before, right? Some viruses apparently do not do antigenic variation. In other words, here's the best way to put this forward. The polio vaccines were developed in the 50s and 60s. We still use them. They work really well against every polio virus that's circulating. The measles vaccine was developed in the 50s. We've been using it. Still works against measles virus. But as you know...
And yellow fever, too.
Yellow fever. But influenza vaccines, we have to change regularly. So what's the difference? Right. And way back when, on TWiV 340, which had a great title, by the way, No Shift Measles. You get it?
Oh, I wonder who wrote that.
No shift.
The man who refuses to get rid of his car and his manual transmission.
No, Alan wrote that. I didn't write that. No. Shift refers to antigenic shift. And it's a play on a word that we don't say on TWiV.
Yes, because this is a family show.
It is a family show, but I just assumed it was you because you refused to get rid of your manual transmission.
Amy, every good title is Alan's without question. Anyway, we talked about what are the constraints on the measles glycoprotein. That's what we're looking at here. And we did a paper where they did mutagenesis of the genome in isolated viruses that could tolerate the mutations. And the only insertions that were tolerated were in the nucleoprotein, the phosphoprotein, the matrix protein. The glycoproteins changes were underrepresented. And so they say, we propose that inelasticity of these proteins prevents sequence variation required to escape antibody neutralization, allowing for long-lived immunity after infection with the virus. This, by the way, is a vaccine that will give you lifelong immunity, as does natural infection. So when I see headlines, we don't have any idea how long antiviral immunity lasts. It really drives me nuts. Of course, we know for some viruses. Anyway, so this paper revisits it in a cool way. That's the neat part of this. So what are they doing here? So antibodies are directed against what we call epitopes on the glycoprotein. It's called the H protein of measles virus. And the epitope, so how big is an epitope? 8 to 12 amino acids typically for a B cell, an antibody epitope, right?
Is that right? Maybe 21.
Yeah, I think
maybe
so,
21.
As much as 21?
Well, the antibody that one of the epitopes that we're interested in is 21
okay.
amino acids.
Anyway, when you are infected with the virus, your immune response makes antibodies against the variety of epitopes. And so they said here, let's look at all of them and see if we can change them, basically. So they collected 30 monoclonal antibodies. A monoclonal, of course, is an antibody against one epitope. And these are neutralizing monoclonals, right? They will block infection of cells. And so it turns out they have 8 epitopes on the measles H glycoprotein that give rise to antibodies. There are 8 distinct epitopes.
Yes, and we thought that there was only 7.
Yeah, they discovered another one in this paper here. And they say, can we change these? And they make a virus with all 8 epitopes ablated. So you can disrupt them. You make probably an amino acid change so the antibody won't bind anymore, right? That's called delta 8. And they write, this is kind of the understatement of the year. These viruses show decreased susceptibility to neutralization by all 30 monoclonals. Really? You think?
That's what she told him to do.
So I want to back up here for a second and give a little measles background, because we've been talking about coronaviruses here for months, where we're talking about a protein, the spike protein, that interacts with a cellular receptor and causes not only the binding to the cell, but also through a conformational change, the fusion. This is not always the case. In the case of measles virus, there are two proteins that together do both those reactions, the binding and the fusion. The protein that does the binding is the hemagglutinin. The protein that does the actual fusion reaction is the fusion protein. And as it is stated in the introduction here, they do so in a concerted process. It's difficult to, they depend on one another to do this. So this is it's both activities in both activities distributed between two proteins, and will make reference to both those proteins because they're both implicated in the rest of this paper. The other thing is that now I get a little confused about this, and maybe you can clarify it for me, because the normal cellular receptor for measles is
SLAM.
There's SLAM.
SLAM and Nectin-4. Right.
Okay.
And the vaccine now binds CD46.
Right. And no longer interacts with SLAM or Nectin-4.
That's
correct? Right. And this Delta 8 dude, so the wild type virus uses SLAM and Nectin-4.
That's right.
The vaccine uses a completely different receptor.
That's right.
And it's sort of, in this context, almost sort of artifactual in a way, right? Because it's an attenuated virus.
Yeah.
Likewise, this thing that they're playing with, where they've knocked out all 8 of these, still recognizes the CD46. What is it?
They've
CD46 receptor. But obviously, I guess because you started with it, it's not going to recognize SLAM or the other thing. So importantly, they haven't created something, that's going to infect humans that evades all the immunity. This is an experiment. This is an experimental system. They've also made the point that they're doing all this in a strain that is
attenuated.
Massively. It's a vaccine strain that's massively attenuated due to a bunch of different mutations in other proteins. And they don't say so explicitly, but the point they're making, I think, here is that, we're doing a lot of engineering and playing around with immune evasion and stuff, but we're doing it in a genetic environment that's totally safe.
Yeah. They're starting out with the vaccine strain of the virus and going from there.
But the reason why one of the reasons why this was interesting is, despite the fact that there's two proteins, that this is still a class 2 fusion process, right, Vincent?
Mm-hmm. It's class 2.
So it's the same process as SARS-CoV-2. So you can make analogies.
Is it class 2 or class 1?
I think it's a class 2. I think
class 2 are flat on the membrane. Class 1 are sticking up. So this is a class 1.
Class 1. Well, so it's the same class as SARS-CoV-2.
Yes. Yep. It's class 1.
So Flavies would be class 2 then, I guess.
So
I always get it backwards.
Were the attenuated strains of measles viruses selected by culture, or were they induced by genetic manipulation?
No, they were selected by passage.
Passage. Okay. So this naturally occurred then.
Well, it tells you that it's a lab-adapted isolate from the throat of David Edmonston. That's why it's called the Edmonston
strain.
I see.
Yep.
Strain or vaccine.
Yep.
How long ago?
1954.
Right.
So 67 years ago.
So this is
1 year
a
less than me.
biological freak.
Yeah, so of course it was not made recombinantly. Recombinant what in 1954?
Yeah, right.
That's it.
By the way, so this vaccine is used extensively. And if you want to know why it does not cause measles, it says attenuation is multifactorial. It arises from acquisition of CD46 tropism, right? The receptor changes, inactivating changes in two proteins that are immune antagonists, and changes in the viral RNA polymerase and in non-coding sequences. All of those contribute to attenuation. And by the way, this vaccine never reverts like the polio vaccine does. It never reverts. I don't know why. Maybe there are too many changes.
There's also no recombination, even though they suggest something about recombination.
Yeah, there isn't any. That's correct.
There's no recombination in negative strands, and there's recombination. So the other thing that is applicable is like the fact that there's receptor utilization change, right?
Yeah.
Yeah.
Yep. CD46 tropism. Yeah. Which is
with these types of evolutionary issues like this never reverts. Polio vaccine does revert. They're often depicted as a 3-dimensional map of evolutionary space where you have, valleys and peaks and ridges. And if you kind of roll the ball into one of these little pockets, it can't get back out again very easily without a tremendous amount of change going to some other pocket. And so that's kind of an analogy that you can use, because we're going to get to that point in this paper where they're talking about the constraints on the evolution of the virus. What would it take to get from A to B? And in the case of the measles vaccine strain, to get from the attenuated version back to something that would cause measles is just too high a ridge to cross. It can't get there.
So we have 8 antigenic sites, 8 B cell epitopes, say. And if you take them all out, you get decreased susceptibility. There's still some neutralization with the monoclonals, but it's decreased. So then they said, let's change one at a time. And if you change any one site, no effect on neutralization. So these are all codominant sites. They're all pretty important. And then it turns out you have to change at least 5 sites to get some resistance to neutralization. So 4, not going to do it. 4 out of these 8, not doing it. So right there you can see. So if you think about, as Alan was saying, this pathway of this virus to resistance, you got to go 1, 2, 3, at least 4. And then as you will see, one of these sites is actually overlapping with the receptor interaction site on the spike. So you can't change that because then it would lose SLAM interaction. So we'll talk about this later. Why the hell would you build a virus like this?
Well,
you wouldn't.
I was going to say, but one who works on picornas might not want to throw too many stones.
Why not? Are there only 4 antigenic sites on picornaviruses?
Well, there's 4 on rhino. There's theoretically 3, although I think Tumakov thinks that there's like 12, but it might not be him. But I think I've read somewhere along the lines that there were 12. And they've been classified from the recent work on Coxsackie A6 as 4 sites.
Okay.
And 2 of those sites are either one is the north rim of the canyon, one is the south rim of the canyon, which in theory, depending on how big if you are a canyon binder, could block or sit at the receptor site. And most neutralization is thought to occur by blocking receptor interaction. So as I said, he who work on picornas may not throw stones.
But most of these are not presumably blocking measles interaction with receptor. They have other mechanisms of neutralization, right? Actually, they don't talk about that, so I don't know what the mechanism is.
They don't really talk. I was going to say, they don't really talk about it. But what was interesting is I think that the ability to switch receptors, right, didn't really affect anything. And we've adapted like 70 to switch receptors.
Enterovirus 70.
Yeah.
For the once again for the listeners, this idea, it may not be obvious, but I find it fascinating. This idea that multiple changes are so difficult to do is understandable if you understand that it's a multiplicative effect. So if in a given round of infection, the probability of getting a change to one of these is, say, 1 in 10,000, which is a reasonable probability given our understanding of how these viruses work, then the probability of getting 2 of them is the product of those 2. So it's 1 in 10 to the 8th, which is what? 1 in 100 million. Mm-hmm. The getting 3 is another 4 orders of magnitude. So now we're 1 in 10 to the 12th. So that's 1 in what, a trillion? And 4 of them would be 1 in 10 to the 16th, which is, I don't know what comes after a trillion.
10 to the
universe
16th.
of stars.
Not real probable.
Yeah.
And I'm sympathetic with this idea of what a stupid thing to do, measles. Okay. But it's an extraordinarily successful virus that's compensated to some extent by how transmissible it is.
Right.
And which I don't know how it does that, but that's maybe another.
Well, and these are all interlocking phenomena, right? I mean, the virus, as we see it, is the end point of a long stretch of evolution. And measles virus, to say that it's poorly evolved, I mean, no, it's not poorly evolved. It transmitted extremely well for a very, very long time. And yeah, it only had one host, but we went everywhere and we took this virus with us. And until the vaccine came along, everybody got measles, right? I mean, you had measles as a kid. That was part of growing up. And so, a bunch of them ended up debilitated for life or dead as a result of having gotten measles, but there was nothing to do about it. And so this was an extremely successful virus. It hit on a great approach. And there's just the downside of that is that it's got this constraint where apparently once you've had it and you get over it, you're immune for life. But that's fine because we were always making more humans, so the virus could keep on going. And then we came up with a vaccine because it didn't count on having infected such smart apes. And so now the vaccine is like having had the virus without having given the virus the benefit of having infected you.
Do we say nanny boo boo?
Yes. Yes, that is exactly what we say when we eradicate a virus.
I guess my point is that what would drive evolution of a glycoprotein with 8 antigenic sites, which would basically make it unable for you to evade antibodies. But you're saying, Alan, it's so infectious that compensates for it.
And I think it's part and parcel. I mean, why does it have these 8 antigenic sites? Well, because it wasn't a problem. And it was obviously evolving other things at the time, like extraordinarily transmissibility. And we always give measles as kind of the example of a highly transmissible virus, where somebody walks into a room and walks out again. And if they had measles, everybody in the room's been exposed.
Yep.
I wonder what the ancestral virus was like. So,
Rinderpest.
Yeah, the Rinderpest. Right. So how many antigenic sites does it have?
Good question.
It's a very good question.
Pretty communicable also.
Yeah,
it's very communicable. Among cows, right?
Yeah.
Not just cows, cattle, sorry.
Cattle, yes.
Yeah, whatever.
The bulls get it too.
Some animal. But, some animal. But yeah, I mean, so I wonder if this is an evolutionarily passed down trait from mother to child virus or
interesting
whatever.
Point, because Rinderpest is the second virus we eradicated, wasn't it?
Right. Or the first, or
it's the first. It's the second. We eradicated smallpox, and just a little while ago we eradicated Rinderpest. And the reason we were able to do that is because the virus never got past the vaccine immunity, right?
Right. So I wonder if this is an that's what I'm saying. Is this an evolutionarily conserved strategy?
Yeah, that and the fact that cattle don't have their wild ancestor has been eradicated as well. The aurochs.
By the way, all these ablation experiments where they remove the antigenic sites, they were tested against mouse antisera. So then they ask, what about human sera? So they get sera from people who have been
cohorts
vaccinated with measles, and they show that, and what they do is they removed the antibodies against the F glycoprotein because those will neutralize infectivity as well. And so they can say, what's the effect of only the antibodies against the H protein? And they show that the viruses Delta 8 with lacking all the sites were also have a 7-fold reduced neutralization with these human sera.
So even if you manage to mutate all of these sites simultaneously and the 10 to the 20 whatever odds that Rich was talking about, it's still going to be heavily neutralized by Yeah.
Yeah, because it turns out there are subdominant epitopes, which they look at.
Right.
And that's probably contributing to that.
Which are non-neutralizing.
Yeah. Now they say, okay, one of these antigenic sites, number 3, overlaps with the sites on the glycoprotein that interact with SLAM and Nectin-4, the 2 receptors for this virus. So they say if you eliminate the B cell epitope, we're going to affect binding. Now the viruses they're using bind CD46, so that's not been a problem so far. But they ask whether these viruses could bind SLAM, and they confirm that they can't. When you change in the delta 8, they won't bind SLAM because the antigenic site and the receptor binding site has changed. So they say emergence of a pathogenic strain with reduced susceptibility to antibody neutralization will require the virus to reconfigure the receptor interacting sites in a way that destroys the B cell epitopes, yet maintains receptor binding. So that's probably a high bar.
Yeah,
I'm guessing.
That's what we call a fitness cost.
It's a fitness
in spades.
cost. Yeah. Says it hasn't happened. So
yeah. So if you do manage to climb over that mountain and get to the other side, you're in much worse shape than when you get there.
Yeah, you're lame.
Yeah.
Then they do a series of experiments where they say, okay, let's make sure that antibodies to F glycoprotein, which as Rich told you is the other glycoprotein in the virus particle, aren't confusing things here. So they substitute the measles virus F gene with the gene from canine distemper virus, right?
I love these. This is so cool.
The morbillivirus of dogs, right? And then because F and H have to interact, they have to introduce changes so that the canine and the human F and H are compatible, right? And so that's a virus called MR now, and which now it's got the human H, it's got the canine distemper F, and they're playing nicely together. The virus is infectious. And they show that, in fact, this is now resistant to neutralization because you've now taken out. So if you use the human sera, they no longer will react with the F from the canine distemper virus. So now the delta 8 would be completely resistant to neutralization, right? Mm-hmm. The last thing they do is, well, actually 2 more things. They find this new epitope by immunizing immunocompetent mice with virus, and then they find antibodies against that weakly cross-react with the H, so they think that there are some subdominant epitopes in there as well. Then they do an experiment which I was confused by because it has nothing to do with anything we've talked about so far. But you may know that people are using oncolytic measles virus to treat certain tumors, and they use.
This is probably why they did these experiments to start with, right?
Maybe. And they use the vaccine strain because the vaccine strain, by virtue of binding CD46 and not SLAM, now selectively kills tumors of certain types. Isn't that incredible?
Yeah.
And that's what Roberto Cattaneo and his colleagues work on.
Well,
so in fact, measles might be really, really smart. I mean, you characterized this as a dumb move, but let's think about this. We've now cultured this virus, adapted it into a vaccine strain, and are going to use it to treat cancer. And if that works, we will absolutely preserve and grow this virus in lots of labs around the world. So it hit the jackpot, right?
Yeah. Dumb like wheat.
Yeah, right. Dumb like dogs.
So here's a question. So if you're using measles as an anti-tumor, aren't you worried about antibodies? People are vaccinated, right? Or they've been infected with measles. They're going to have these antibodies. So they made, and as Rich said, it's probably why they made this virus. They have delta 8 with the F protein of canine distemper, and they give antibodies, anti-measles antibodies to mice, or maybe the mice are immunized. I don't remember. But basically, they infect tumor-bearing mice who have antibodies to virus, and the virus has no problem getting to the tumor and reproducing because it's not neutralized by any of the antibodies.
Right. So now they've created an oncolytic vector that they can use in people who've been vaccinated against measles or had measles. The senior author on this paper is Stephen Russell, who is part of those trials, who we had on TWiV previously. Let me see. I've got it up here.
Did
TWiV
we really?
390. Yep, TWiV 395. It was the Virginia Tech ASV meeting. He was a keynote speaker.
That's right.
He gave a brilliant talk, and he was great on TWiV. 395, The Cancer Thief, June 2016.
Right. I forgot the connection there. So yes, this is probably why they did the paper, to do this experiment. You're right.
Yeah.
And the
kill
F
two birds
protein,
with one stone.
this is brilliant. So people are going to have antibodies to H and F. So they get rid of all the H epitopes. They put in F of canine distemper. They make this fancy virus where the F and the H talk to each other, and it will still infect the tumor.
And it can't get back to any of the previous, like it's not going to turn into measles by any chance.
It's not going to turn into measles.
Nevertheless, if you broadcast this episode to the conspiracy theorists, okay, they will spin it into something that sounds horrific.
They'll never find it. Look, the only reason they found the Dashak episode is because it was Dashak, right? And you just do a Google for Dashak, which they're doing because they all think he's evil. Oh, look at those cool airplanes. VTOLs just went up to Hudson.
Oh, cool.
You know, with the propeller facing up.
The Ospreys?
That's cool.
Yeah. So I don't think they'd find it, Rich. Yeah.
I don't know. They're going to be looking for you pretty soon, Vincent.
So they do a great summary here. Let me just do this. So by engineering the surface glycoproteins, we elucidated a number of factors determining the remarkable antigenic stability. First, there are numerous immunologically co-dominant antigenic sites on the H and F. Second, antibodies to each of these sites can neutralize infectivity. Third, measles with even a single site left remain fully susceptible to neutralization. Fourth, the receptor binding surface is itself an antigenic site. Hence, measles cannot escape their susceptibility to neutralization by human serum unless they also lose their tropism for the pathogenicity determining receptors SLAM-F and Nectin-4. But it doesn't care. The virus cares not because, as Alan said, it can affect so many people. Who the hell cares? Given that a minimum of 5 sites must be disrupted, the probability of it happening naturally is vanishingly small. And disruption of fewer than 5 would confer no selective advantage on the virus, making stepwise evolution an unrealistic pathway. However, even a measles sensitive to anti-H antibodies would still be neutralized by anti-F, and even more critical, would lack SLAM-F and Nectin-4 tropisms required for pathogenicity.
No matter which way it turns.
And the requirement for HF interaction is an additional constraint on evolution. This presents a stark contrast for other viruses such as influenza and corona, where a much smaller number of amino acid substitutions at key positions within a single virus-embedded glycoprotein can cause escape from sera. We therefore conclude there is a near zero probability for the accidental emergence of a pathogenic measles virus capable of evading vaccine-induced immunity. That's just an amazing statement, isn't it?
Yeah.
Very definitive.
It's in such a specific evolutionary space that it can't get out.
So having the two proteins is part of it too. Yeah, that's quite interesting.
Do we know why measles is so transmissible? I mean, here we've been having this debate throughout the pandemic about the difference between droplet and airborne and what's aerosol, blah, blah, blah. Okay. But in measles, there's no question, but that it is airborne. Got to get the terms right. Airborne.
Yeah. It's in the aerosols.
Somebody with measles walks through a room and you can come in there long after they're gone and you got measles. Okay.
Yeah.
Because it'll hang around in the air. Do we know why? I mean, it's an envelope virus.
I think that this is just my bias, let's say. So it's probably wrong. I think that, so the fine droplets that travel very far, right, the aerosols, so-called, they're small, and so they physically can't have a lot of virus particles in them. But I think for measles virus, it's so infectious that if you whatever particles are in those small droplets are enough to infect you.
Okay,
Whereas
so you don't need a big dose.
I don't think you need a big dose. So with SARS-CoV-2, most of the transmission is with the bigger droplets, right? Sometimes the little ones happen, but I think that's unusual because there's not a lot of virus in them. And I think you're in situations where you're sitting inside for a long time and there's a lot of smaller droplets that you're inhaling. Whereas, for measles, no matter where, a very quick encounter, the little droplets are good enough. That's what I think anyway.
So basically what you're saying is that if you have 100 particles of measles, 90 of them are infectious wild type virus, 10 of them are not. Whereas for SARS and flu, the ratio is the opposite.
That could be part of it. Yeah, that could be.
Well, that is it. Otherwise, that's the essence of it. Yeah, exactly. The rest
How
is
can
just
you tell
fluff.
me what I'm thinking?
I'm not telling you what you're thinking. I'm just stating a fact.
That was what you just argued.
Could you repeat it? Could you repeat it? So 100 particles.
Okay, so
90 of measles are infectious.
Right. So it doesn't matter if I go through big droplets or little droplets, because the probability of that droplet having an infectious particle that can
is really high. Yeah,
is very high.
And now for SARS-CoV-2, it's maybe 10.
It's the opposite.
It's 10 out of 100.
Right.
Yeah,
it's 10 out of 100.
Okay.
So the probability of a small droplet having an infectious particle, the probability is significantly reduced. Therefore, I go through a big thing because I need to have all 100 there to have at least 10 that can launch the infection.
Okay.
Infectious defined as you, the human,
Getting disease.
Getting disease
infected.
or getting infected.
Yeah,
Not only
whatever.
that, the smallest particles go deeper into your lungs. So,
But then
much
your
better
lungs
chance of
are a
retaining
dead
it.
end.
So these little, really fine aerosols, what are they, like a micron in diameter?
We have this, I think, pick coming up. They are less than 5 microns. That's the arbitrary
less than
cutoff.
5.
All right, so a micron is 1,000 nanometers, right?
Right.
Mm-hmm.
And a virus, so I think measles is like 30 to 40 nanometers. So it ain't a big space, right? I would say measles and coronas are probably on the order similar sized diameter, right?
I would think so.
But
Corona's maybe bigger. It's got more RNA.
Now, if you were talking about a giant virus, which is 10 times bigger, then the physical size of the particle could also limit the ability of fine particles to transmit it.
Could be.
Well, when was the last time you saw a giant virus floating through the air?
Why did I
I've
think
only
you were
seen them
gonna-
in the water.
You wouldn't see it at all.
I was going to say, but I've only seen it isolated from
They're in
water.
aquatic.
Yeah, I know.
Yeah.
Why did I know you were going to say that? I was
I'm
just
not
making,
clear.
I'm just making an academic point. It doesn't necessarily mean it happens in the real world. And I know you say, why bother to say it at all? But I'd like to do these things. Okay. So in this case, the size of the particle, but yes, it's the infectivity. So someone needs to do the particle to PFU ratio for SARS-CoV-2.
Well, the problem is that PFU doesn't necessarily translate to infecting a human.
Infectivity. Yes.
Yeah, it may not be one.
This
one particle.
goes back to some of my basic virology lectures where I compare infectivity, compare TCID50 with egg infectious dose with plaque forming units, and you get different numbers depending on the assay.
Okay.
Yeah.
Okay. The assay in this case, we got to do a plaque assay on humans. Okay?
Yes, you're right. You're right. Which never going
By
to
the
happen.
way, any conspiracy theorists who are listening, who are going to spin conspiracy out of this, I should point out for your benefit, if you want to decode the secrets of what Vincent is saying, you actually need to get the code by listening to his virology course first, and then proceed through TWiV, and then you'll understand.
So when someone has measles, how many particles do they shed per breath?
Lots.
No, that's not.
I don't know. I don't know offhand.
Sorry.
Maybe it's known, but I don't know.
Because maybe that's the difference between SARS-CoV-2.
That could be an element of this. Yeah.
This paper is even better than I thought it was.
Yeah, spun off this whole discussion.
Thank God I'm in charge of picking the papers.
That's right.
But don't you ever find that sometimes after you have, you could read a paper and you think something, but then you sit down with 4 people and you talk about it and you actually get more insight?
Right.
Yeah.
Hey, maybe we should do a podcast based on that idea.
About that.
Good idea.
Nah, it didn't ever work.
No? Okay.
It'd probably only get about 10 or 20
because you have to have the whole conversation planned out. And you can't do this spontaneous.
You got to script it.
It's all got to be planned out. It's got to be scripted.
Okay.
Do we know the repertoire of tumor cells that this virus can infect?
Anything that has CD
46.
So it's been tried in people in multiple myeloma, right? A B-cell tumor. That's the one. But I think it's also being looked at for
and gliomas.
Gliomas as well. Yeah, I think there are a variety of
solid tumors. Yeah.
I believe it's been used for brain tumors.
So here's the thing, Dixon. There's the trial that was done a couple of years ago. It was published. It was in 2 women who had advanced multiple myeloma, refractory to any other treatment, right? There's nothing left. So they decided to try this. They gave them intravenously 10 to the 11th particles of measles virus, vaccine virus, right? Infectious. And they had not just B-cell tumors, which means, in them, but they had physical lumps on their heads, right? And it hurt. And the one woman said as soon as that virus got in her, she felt the pressure being released on her forehead. And eventually the lumps went away because the virus was killing them. And I think one of the 2 ended up in complete remission.
Yes.
Right.
That's all in that episode. It probably answers some of your questions about what other therapies are being done.
Oh, did he talk about it in that episode, Stephen Russell?
Yeah, I think so. Yeah,
we have to link that. That's a good one. I had forgotten about that.
Yeah, I wasn't on that one, so I actually listened to it.
Stephen Russell. I remember he had his arm in a cast.
That's right.
Because he fell off his bike.
It fell off his bike. Yep.
But I told people, I said, Steve, I'm sorry I had to twist your arm to come on TWiV. You get it, Dixon?
I got it.
All right, let's do one round of email so we get through some of these. Rich, can you take that first one?
Sure. Hang on.
Monique.
Monique writes, love the show. Invaluable information and scientific data review feeds my rational scientific mind to combat all of the other noise out there. I am a 55-year-old Canadian female. I took the first shot offered, which was AstraZeneca. I determined it would get our numbers down, help the pipeline for other people, to get Pfizer for my 3 daughters, 19, 21, and 23. Now the amount of AZ is limited. AstraZeneca is limited. And some provinces are stopping the use of AstraZeneca. The debate now relates to which vaccine people whose first shot was AstraZeneca should get as their second shot. In British Columbia, where I live, we apparently will have the choice of AstraZeneca or Pfizer. Any advantage on which one people in my situation should pick? By the way, I had all the side effects possible with AstraZeneca: headache, chills, fever, nausea, and vomiting. Survived, and a small price to pay for getting this virus in control. I have seen the research that side effects are greater with a different second vaccine. Pfizer and Moderna have increased side effects with a second shot. AZ has less side effects with a second shot. Thanks in advance for your guidance. This is similar to a letter we had a couple of days ago. I personally don't have any data. I would say, number one, that I don't know of trials other than, actually, there's an ongoing British trial that mix and matches as AstraZeneca and Pfizer. I don't know if there are preliminary results from that. I don't know how that's going. But there will be data on that eventually. But I think you want to get your second shot first if you can. The only other thing we know is that the trials with AstraZeneca say you can do 2 shots of AstraZeneca and you're okay. So those are the data. Theoretically, I don't think it makes any difference.
So the results, preliminary results of that mix and match are out. They have 600 people in Spain vaccinating people with both AstraZeneca, and Pfizer produces a potent immune response, as they found in a study in Spain. Okay.
Anything about side effects?
Let's see. Blah, blah, blah. Side effects, prime and boost. Let me search for side effects here. Here we go. Giving people first and second doses. No, nothing about side effects here.
Which probably means that there's no big deal going on.
Here we go.
So,
last week, a UK study called COMCOV, which analyzed combinations of the same two vaccines, found that people in the mix and match had higher rates of common vaccine-related side effects, such as fever, than did people who received 2 doses of the same vaccine. In the Spanish trial, the mild side effects were common and similar to those seen in standard regimens. None seems
deemed
severe, so you could go for an mRNA vaccine if you want. It's a hard decision because of the clotting issue with AZ. You should leave it up to the patient, right?
Yeah, and consult with your doctor is always always the bottom line advice. But I think Daniel would say the vaccine you want is the one you can get, right?
Yeah. But she says she has a choice. So
yeah, I understand from this letter that she's expecting to get a choice.
Yeah. By the way, I really like that she feels her side effects were a small price to pay to get this virus in control. What a great way to look at it, right?
Yeah.
Yep.
Thank you, Monique. Really appreciate it.
I would probably favor just getting the AstraZeneca. I don't know if there are data on who gets the clotting. Like, is it the case that if you didn't get it with the first dose, you're probably not going to get it with the second? Like maybe there's some genetic predisposition, or is it more of a Russian roulette thing? Like, do you want to fire another shot of that if you didn't get it the first time? It's an extremely rare side effect. My bet would be on some kind of a genetic or HLA linked thing. And if you didn't get it the first time, you're probably fine.
Is that the age group?
Right. My other thought on this is we have clinical trial data on 2 shots of the AstraZeneca and like full scale. The only trial data we have on the mix and match is now from these smaller trials in UK and Spain. Probably fine. I don't know. Under the circumstances, I'd probably get the AstraZeneca. But
I'm seeing reports.
I don't think you can go wrong.
That it's mainly first dose, the clotting issues.
Yeah, that was my understanding.
Yeah.
So if you survive the first dose, you're probably okay.
And it's mostly premenopausal women.
Right.
She's probably not in the right age group.
Maybe. Yeah, there have been. She could potentially, but it's probably not an issue. And anyway.
Okay.
Yeah, up to you, Monique, is basically what we're saying.
Yep.
Talk to your physician.
6 to 1, half a dozen the other. Let us know how it turns out.
Yes.
Dixon, could you take the next one?
Yes, I can. Mary writes, "Hi to all at TWiV. Greetings from Ireland on a typical summer's day, cloudy with a chance of rain and sun. Thanks so much to you and for all your amazing work. My mother-in-law, who is highly religious, Catholic faith, how unusual, excuse me, is refusing to take any vaccine as she has been told that the aborted fetuses that have been used during vaccine development. I would be very grateful if you could advise us on what to say to her. Regards, Mary."
Just quick, Dixon, if you could maybe move your mic a little bit. I think your whiskers are brushing against it.
Yeah, there we go.
Whiskers. Wow, good. What an ancient term, whiskers.
Whiskers, my whiskers. Okay.
Wow.
Should I read this again now?
No, you're okay.
You're okay. What do we say to her?
Well, I'm unclear where the aborted fetus cells come from an mRNA vaccine.
We can parse this out several ways.
Yeah.
Okay. First of all, the aborted fetuses that we're talking about come into play because some of the cells that are used in either the experimentation during vaccine development or, depending on the vaccine, during the production of the vaccine, were made from aborted fetuses. We're talking at least 30 years ago. Some of the cell lines, some of them are 50 years ago and have been maintained in culture ever since. There are no, it's not like fetuses are aborted currently actively for the purpose of making vaccine. We're talking about established cell lines that probably are so removed from the initial tissue that they're unrecognizable. Nevertheless, they did originally, years ago, come from aborted fetuses. Some of those cell lines were used for preclinical studies to produce or experiment with the mRNA vaccines, but no cells are used for the production of those vaccines. So you can be confident that if you take a messenger RNA vaccine, you're not taking something that was produced on cells from an aborted fetus. In the case of the viral vectored vaccines, and that includes the adenoviral vectored vaccines, Janssen/Johnson & Johnson, or Gamaleya, or AstraZeneca, those vaccines are produced on these cells that were their cell lines that were made decades ago from aborted fetuses. They are still used to produce those. I should say that the Pope, this is no joke, has ruled that this is acceptable. Okay. And he has told his flock, okay, that the use of these tools to produce these vaccines, the vaccines themselves are important enough in saving lives so that overshadows any potential downside to vaccines produced on these cells from aborted fetuses. So that's what I would tell Grandma.
And I would also point out that in January, I was just Googling to try and see if I could figure out which vaccine it was, but Pope Francis and former Pope Benedict both have received COVID-19 vaccine. So I don't know if it's whatever's approved in Italy. I mean, technically they're in the country of Vatican City, which is its own nation, but it's probably whatever's approved in Italy is what they are both receiving. And Pope Francis has also made the statement that ethically this is acceptable.
Does Vatican City have its own FDA?
I don't know.
I'm just kidding. I doubt it. It's just like a bunch of buildings, right?
Yeah, it's just some buildings in Rome.
Yeah. So, Amy, I wondered the same thing for the mRNA vaccines, but if you look at the papers, they put the mRNA into 293 cells to make sure it made protein, and there you go. That's the catch. Some people don't even want that.
Well, I also have a problem with aborted fetuses. So the way it's stated, it's sound, and the way kind of it was described by Rich, unfortunately, I don't think he meant it this way, but it sounds like the fetuses were aborted so that we could generate cell lines, and they were not.
Right.
Yeah.
Aborted. These tissues would have been discarded for various.
Right.
They had various genetic whatevers or whatever reasons, and then these tissues would have been aborted. These tissues would have been discarded.
Yeah.
Otherwise,
I don't think that matters to some people, though, right?
Well, I think it does, because I think like the idea is today is that you're paying women to abort their fetuses so you can make the cell lines.
Nobody has done that.
No.
Yeah, that's what I'm saying is like, that's not what happened. What happened was these tissues would have been discarded, just like, other tumors would have been discarded.
These were,
you could just dissociate them and ask in the 50s, could I grow a cell line?
Right.
Which is basically the way I understand, like, they found the HeLa cell line was that they took all these discarded tumors, they dissociated them, they filled their incubators, and lo and behold, one cell line grew continuously.
Yeah.
Well, I think some people, so Mary's grandmother might say, oh, okay, that's fine. I didn't know that, because I think a lot of people don't understand what we've just explained. But others have written to me and said, even if the 293s were used to verify that the mRNA could be translated, I don't want anything to do with that. And that's your choice. But I think a lot of people are not that strict about it. So,
And I mean, my understanding is that Catholics should probably go with what the Pope has said. But not having been raised Catholic, I'm not going to go.
I always ate meat on Friday, man.
Okay.
But I'm not the shining example of, no, we grew up as Catholics, and as soon as I got out of the house, I started eating meat whenever I wanted to.
The other thing I would say is simply that I agree with you. It's your choice. Understand what the issues are clearly.
Yeah.
Understand what the science is, what the ethics are, and then make your choice. Understand that the flip side of choosing not to get vaccinated is getting COVID. Yep. Okay, and that's not good.
Right.
And I also just want to point out the intense irony of this devout Catholic choosing to exercise choice over an issue of medical individual action. And I would just like to maybe suggest that she pray on that idea a little bit.
Well, I think you should just listen to Daniel, where he tells you, what was it, like 2 weekends ago, where Daniel had the story about the wife who got vaccinated and the husband who did not, and then like died 2 weeks later.
Yep.
Right.
Was that, okay.
It's your choice.
You're playing with fire.
You are.
You're going to get burned at some point.
Alan, can you take the next one?
Sure. Hillary writes, listening to TWiV 757 this morning, clear and sunny at about 50 degrees. I was amused to hear the author of your featured paper refer proudly to actionable results that might really be actionable in the old and still primary meaning of the word. The idea, if I understood it properly, was that some people are shedding a whole lot more SARS-CoV-2 than others. So if you could catch them and stuff them into quarantine, you could have a huge effect on the spread of COVID-19 while more or less ignoring the ones who weren't as generous with the virus. So you'd be discriminating among people and constraining some more than others. And some would be bound to object or try to evade the restrictions and wind up in court as a result. And that is precisely what actionable asserts. The action in question is legal action. I know language does change and that more and more people do use the word to mean something we're glad to be able to do something with or about. But in 20 years as a freelance editor, I never had an author object to finding other words for the concept when I pointed out the Snicker factory. Why boast? This will get us sued. With viruses, I'm a total innocent and believe everything you say. But with words?
I like the Snicker factor. That's great.
Actionable. I didn't know that. I didn't know that.
No.
Yes. Something that's actionable is something that can be the subject of legal action. I have occasionally slipped into using actionable to mean something that you can take action on, but it is correct that the original meaning of it is I slipped and fell. This is actionable. Yeah.
Amy, can you take the next one?
Sure. Soren writes, hello, TWiV folks. When the immune system is exposed to a new antigen, it produces new antibodies. Those antibodies are proteins. Where is the template for producing those proteins? I don't think the immune system can create new DNA sequences for antibodies. Even if they do, I assume the immune system doesn't insert those sequences into the genome. It seems like the body has another way to remember and produce antibody proteins, and I can't even begin to guess how that happens. So to produce antibodies, you have 3 regions. Now, I will confess, I've never actually taken an immunology class. So this probably I'm going to get wrong, but dumb it down a bit. You have 3 regions and they undergo random recombination. And then they make a protein, then they make RNA and make a protein, and then it's produced as an antibody. And so, no, the immune system is not inserting those sequences into genes. The genes are there and they're actually deleting sequences from any other way. And since it's it's in the genome, it's remembered because the DNA cannot get back the sequences that is deleted and degraded. So then the cell reproduces, and now the genome is always going to have that protein encoding sequence within it, and it's going to make more and more protein and reproduce.
And don't forget somatic hypermutation.
I said I was doing this simple way.
I just want to jump in here and say, Soren, actually what you're describing here is, as you know, this preposterous idea that the immune system is somehow rewriting the genome is what it is doing. Yeah. And yes, you're right. It's preposterous. And when people first discovered this, they were like, what? The the cells of the immune system are rewriting their own genomes. And what Amy was just describing is the mechanics of that. There are, in fact, segments. There's a whole locus, a whole set of loci in your genome that have evolved to shuffle themselves around in the cells of your immune system. And so, yeah, that's exactly what happened. The cells of your immune system rewrote their genome to make the antibody. And then there's a lineage of those cells that has a different rearranged genome. And they're circulating around with this reprogrammed genome, and they're living throughout the rest of your life. So your genome is being rewritten in real time every time you encounter a new antigen. And it's really bizarre, and it baffles common sense. You're absolutely right to find this preposterous, and you should listen to the Immune podcast and find out more about how weird the immune system gets.
My complaint is that it's automatically assumed that there's something inserted.
Well,
I'm inserting something, but I'm not. I'm deleting stuff.
Yeah, but then you're putting it into the locus to.
It is the locus. I'm deleting the locus. I'm deleting. Yes. I'm making a 3 kilobase locus 1 kilobase.
Okay.
I also should say that there are enzymes that then mutate the DNA and change bases so that you get different proteins
and then rewrite it further.
And then the ones that are really good binding to the antigen are selected in the lymph nodes. So it's just amazing.
Yeah,
it is amazing, but I don't see why we need to make it more complex and scary for people. We should just say what it is. I think it's
okay. I believe in complexity. I think people can handle it. I think Soren basically is tongue-in-cheek. He probably knows all this. He wanted to see what we would say.
I don't know.
But if Soren didn't know this and has intuited
that's a great question.
The antibodies are proteins. So what's the template? Can the immune system create new sequences? And my answer to Soren, if this question is sincere, is yeah, it can. Isn't that freaking bizarre?
It's a great question. When I saw it, I said, this is brilliant. Yeah.
Yeah.
All right, one more. Sue Ellen writes, I'm sure I'm not alone in saying that the non-virus arcs and digressions are some of my favorite moments on TWiV and your other podcasts. I think those of us who enjoy those moments, such as the recent one labeled the Panhandle discussion, just don't write in to say how much we like them. So you are hearing only one side, the cranky pants out there who want their virology neat in a glass, not shaken or stirred. I think I've said before that so much of what makes the Twix podcast so very enjoyable is the fact that you share all kinds of personal information with your listeners. I know about Vincent's garden, Rich's 2, Michelle's love of Michigan football, Elio's obsession with mushrooms, Dixon's fishing expeditions, and I enjoy all of those digressions. I'm especially tickled when you all get totally geeked out by something, like the time you all spent several minutes using a New York Times link to figure out how far down the line to get a vaccine each of you would be. I remember that. So please don't take it to heart when people write in to complain. There are plenty of us out here who love the weather reports and the digressions. We just don't bother to write in about it because we can't imagine that anyone else out there isn't enjoying them as much as we are. Mm-hmm. Sue Ellen. Thank you, Sue Ellen.
Thank you. Appreciate that.
And I just wanted to insert a digression here. I get tons of emails from China. They say they want to sell me valves and tires and tubes, anything in huge quantities. We can process your order for 10,000 tire valves. I don't know why I get these, but I got this one yesterday. Dear purchasing manager, hello. We provide you with China Qixing Zhongwei inactivated coronavirus vaccine, Virocell, disposable syringes, new cap detection boxes, masks, and other anti-epidemic supplies. Best wishes. Zhejiang Canyue Medical Technology Limited, Zhejiang, China. I thought this was so funny, right?
Well, did you buy some? What's the price?
I don't know. I'm not sure. I mean, if I responded, what would happen? That would be funny. Yes,
you'd get 10,000 other emails is what?
Yeah, right. Then,
you'll end up as the owner of TikTok.
I mean, I think you would probably get fleeced.
He's not getting the P card, so it doesn't matter.
Yeah,
is that a tire brand?
In this email, I can confirm that Zhejiang, China, the city, exists.
Yeah.
The rest of it, I wouldn't bet any large money.
As I said, I'm in charge of the credit card. He's
It's
not
funny
getting
that-
a number.
It's just funny.
Don't
I
worry.
usually get solicitations for, washers and bolts and all kinds of esoteric stuff in huge quantity, but the vaccine, then it says Virocell on top of it. I just pisser. All right, time for some picks. Dixon, what do you have for us?
You're muted, Dixon.
I will unmute it. I'm unmuting. I just bought a book for my grandson called Ants, Workers of the World. At least I thought it was for him. I'm almost afraid to give it to him now because it's got such intimidating pictures in it. The pictures are phenomenal, by the way. These are wonderful, three-dimensionalized pictures of ants that look like they could consume Cleveland. And when you're up close and the head of an ant is the same size as your head, it's really interesting to look inside the mouth of an ant. Most of them are workers, and he's got some queens in there also. I looked through virtually every page. At the end, this is not a kid's book. This is definitely not a kid's book.
Well, this is
I'm not so sure.
book.
Yeah,
But
Porter would love this.
It's beautifully, beautifully done, and it's an
looks
Abrams
great.
book. Abrams is an art book company, and Abrams actually produced this as an art book,
Yeah.
and it is fabulous. That's all I can say. These are the best pictures of ants I've ever seen.
I guess you don't want the Kindle version, huh?
That's right. They do have a Kindle.
So it's mostly it's photography mostly.
Yeah, it's got a sparse amount of words, but Edward Rice was the photographer and Eleanor Spicer was the wordsmith. But it really gives you a very unusual picture of life at that level. And I'm going to go back and look at that book a lot because I can't believe that that's what it looks like. If I was that size, I would be totally freaked out.
So you're
I
not
know
gonna,
some people who are getting this for Christmas. This is awesome.
You're not going to give it to your grandson, Dixon?
Well, I'm going to let him look at the cover, and if he wants to look at the rest of the book, that's fine.
These are gorgeous.
They are. They really, really are. And then they're not colorized either, Alan. Those are the real colors of the ants.
Well, no, yeah, I'm getting that. That's
I mean, because a lot of the photographs of ants that I've seen in the past are scanning electron micrographs,
Right.
which are then colorized, and it doesn't work.
no, these are macro photos. These are the real deal.
Yeah, but these are
Beautifully lit, beautifully shot. This is hard to do too.
no kidding. No
Wow.
kidding. No kidding. This guy is
Okay.
originally from North Carolina, by the way.
Cool.
Very cool.
Thank
Very
you.
nice.
Thank you, Dixon.
Thank you, Dixon.
Porter, I don't think listens to TWiV.
You're welcome.
His birthday is coming up, and I just ordered it.
Nice.
Oh, my goodness.
Nice present.
Hey,
Yeah,
Vincent,
great.
didn't you have something similar on TWiV with Nels and the guy from Rockefeller?
Yeah, Daniel Cronauer
Who
has a
does
book,
ants?
Army Ants, and
Oh, really? Oh, that's right. That's
Daniel
right. You did
goes
mention
to various countries to work on Army ants. He takes pictures, and the book is gorgeous.
Wow.
Yeah, also very
Wow.
gorgeous. Amy, what do you have for us?
So Let the Record Show is a social history of HIV during the late 80s and 92, the social movement that occurred in downtown Manhattan, and how they got better healthcare for women who are HIV positive and better healthcare for really opened up the idea for acceptance that it was not a gay man's disease and stuff. And it just is because that pandemic is still ongoing and we seem to just take it for granted that it is been done with, or, it doesn't really make the news anymore. So she and her husband, who's a film producer, put this book together to highlight, the social ways that we combated the biases and problems of that and how that might be applicable
Cool.
to what comes up today. Maybe it's not anti-gay, it's anti-Asian or whatever.
Nice.
Excellent.
This is good.
Cool. Rich, what do you have?
I have another TWiV pick from my TWiV feed. My TWiV pick feed, Grant McFadden. He's one of several of these. This is a Washington Post article called QAnon Resembles the Games I Design, but for believers, there is no winning, by a gentleman named Reed Berkowitz. who apparently designs these games that Alan may have some familiarity with them. He talks about games all the time. I don't. These sound like online games that are multiplayer.
These are like mixed reality type games where they're augmented reality. I don't think he was involved in Pokemon Go, but it's kind of that sort of thing where you interact with the electronic part of the game, but also with the real world, and you collect clues from the real world in order to play the actual game.
And you put clues together, and there's a goal, and when you reach the goal, you get a prize and all that kind of stuff. And this person, the author, programs these sorts of games. And when he sort of started poking into QAnon, he realized that the mentality is exactly the same. Okay, that hooks QAnon believers. The difference is that there's no goal, okay, and that it causes problems. But I learned a new word in the process, which is epiphany, okay, and that's where it's derived from a phenomenon called apophenia.
Epiphania,
which is the tendency to perceive meaningful connections between unrelated things. And in psychology, apparently, there's a human tendency to do this, right? Try and make connections. Yeah. Makes perfect sense.
My wife has a lot of patients who do this. Yeah.
Right. Well, and in the extreme, this is one of the it's diagnostic of schizophrenia, or at least the beginnings of schizophrenia. And, I get it. Is this a coincidence? Or is it not? And so I thought that this provided, we all the time wonder, how can people get sucked into this? All right. And this article is a really good insight into that, okay, because it puts it out, as a, comparing it to this sort of game thing, and you can kind of understand it if you've played any game similar to this, how you could get totally sucked in. So,
wow,
there you go, apophenia.
Excellent insight. Terrifying.
I got sucked into this article. Marjorie Greene isn't here to legislate; she's here to live stream. Go read that. Oh my God. Oh boy.
Well, so once you read this and you understand, is there like the deprogramming paragraph? No, I'm serious. Like, do you learn how to deprogram these people? There's people who like joined cults in the 70s and 80s and stuff, and they came out and they had to be deprogrammed.
Yeah, it's going to be something similar. And I mean, when this happens pathologically, when somebody suffers from schizophrenia or some related, there's an actual biochemical neurological thing, and they're seeing meaning in things that aren't there. And if you think about it, we all do this. We all look for patterns. It's just a fundamental human trait. And so when it derails, and then you're looking for patterns that aren't that you see, but that nobody else does, then that's a problem. And these folks, I think Amy's analogy is spot on. It's like a cult, and they are neurologically probably healthy people, normal people, what we would call normal, who've been led down this path, and who then are going to have to reconstruct their identities without this. And yeah, that's going to be a whole process.
Well, I'm not sure, Alan, that you're being very generous by saying these are normal people that were attracted to this.
I think they started out as normal people.
No, you and I are normal people, and we're not attracted to that. And a lot of other normal people are not attracted to this.
But I'm not from the same background they are.
Really, I just wild people.
I'm hesitant. I'm hesitant to say that I couldn't be in that situation.
No. We won't allow you.
I would like to...
the commune will not allow you to escape.
I would like to announce that I have joined the cult of virus.
Yes. Yes.
Well,
we can deprogram.
This is all true.
Well, in thinking
got a vaccine for that.
In thinking about your issue, Dixon, I was thinking about this morning. I have to remind myself very consistently that I live in a bubble and have my whole life, a very small bubble of hyper rational people. Okay, and we're talking nerds here. And my experience with the population at large is really limited. And so I think it takes all kinds, as they say, and the world is a pretty heterogeneous place.
Well, I think,
but of course,
I think living on the East Coast is a huge bubble. I think, if anything, we should have gathered that from the results of previous elections, that we live in a bubble. You live in a bubble if you look at the economics. Like, a lot of people don't have the monetary means to go on a physical vacation. Like, you and Marlene travel a great deal. And most of the country thinks that going on vacation is watching football and drinking beer on Sundays. It's a vacation. It's a vacation from their lives. So, I wouldn't say that those were not normal people. They're just, we live in one little bubble.
Speaking of East Coast mentality, Amy, you know how they populated California? They lifted up the country at the East Coast, and everything that was loose rolled west.
That's good.
That's good.
Many of them drowned.
But, I think it's true, like, that there's geographical bubbles, there's economic bubbles, there's cultural bubbles.
Sure.
I mean, people write in all the time when we do this live stream thing on Wednesday night. They're like, I wish I could go to Zabar's. I could, and then I meet people walking down the street and they're like, what's Zabar's? It's a New York Jewish bubble thing, right? Not everybody knows what Zabar's and a schmear is.
I know, and I'm not Jewish.
You live in New York.
You live in New York.
And you're surrounded by Jews. I mean, you lived next to Saul for 30 years. There's got to be something that rubbed off. Besides,
you're still chopped liver.
I am chopped liver. It's fine. It's not a problem. You're right. Saul Silverstein was my neighbor here at Columbia for many years, and he had the book. What is the book?
That's right, the Book of Jewish Humor.
Leo Rostam.
He also he had that too, but he had. I shipped it to him. He would not let me throw out.
It's the joy of Yiddish.
Dictionary of Yiddish. And Steve, who was, I don't know, non-religious or whatever you want to call it.
Steve Goff.
Yeah. He printed out an entire page and stuck it to the bulletin board outside your office that said the 30 Yiddish words everybody needed to learn, like schmuck, bubkiss,
Schmear. Is schmear a Yiddish word?
Yeah. Schmear means you spread. Thank you.
Yeah, Saul would periodically bring out the book and tell me a word or two here and there. Yeah. I always say, "What's a mensch?" I said, "What's a mensch?" And he brought the book out. He said, "I'll tell you, it's a better way is to give you an example. Elvin Cabot is a mensch. Now, Elvin Cabot was a faculty member here, and he was a nice guy.
He was a mensch.
He was a mensch.
He was a mensch. And what are you talking about? Every other word out of your mouth is, you're giving me surus, Amy.
Saul told me surus. That's a great word. I really like surus.
Way, but I can afford business class now.
I really like surus. Okay, Alan, what do you have for us?
Okay, I have a site called LibriVox. And if you're into audiobooks, my wife has gotten really into audiobooks because she's got a commute now, and she puts them on in the car, and she's been pestering me to start doing audiobooks because she says, "Oh, you could narrate better than some of these people." So I started looking into it, and I stumbled across this. This is something I can really get behind. It's free audiobooks of texts that are in the public domain, and there's a charming little community of open source hippies behind this who have decided they're going to take texts that are in the public domain, make audiobooks of them, and release the audiobooks into the public domain. And for those who are not up on copyrights, public domain is the most extreme version of free. Like, this is so free, you can do whatever the heck you want with these. So if you want audio for something, just fine, take it. And so if you want to listen to free public domain audiobooks, you can go to their catalog. If you want to, it's basically anything that was published before 1925 in the United States has fallen into the public domain as a text. And the LibriVox volunteers are working their way through. They've got now a dramatic reading of The Great Gatsby going on, so that should come out sometime soon. That just went into the public domain, but then they've got stuff going way back. If you want to listen to all your classic novels, you want to hear somebody reading Pride and Prejudice, don't pay for the audiobook on Amazon. Just get it from LibriVox for free.
Oh, this is amazing. This is quite a catalog.
And they've got over 15,000 of them now. Yeah.
Wow.
And if you feel like reading these, I mean, everybody on this podcast has a good mic and an audio setup, and you can go to their forum. And it's, as I say, it's this charming group of open source hippies who are doing this.
Oh, cool. Neat. Yeah, you should record them, Alan. You have a good...
I actually am. I am on the forum now, and I'm doing a couple projects with them.
Is that why you grew the mustache?
No, I've had that for decades.
Oh, okay.
But you could also do it for get paid. You could do voiceovers.
Yeah, I have heard
right.
that. I've looked into that a little bit, but this is more fun.
You'd need a booth. You'd have to have a soundproof booth, but you could.
Well,
do
you
that.
have to go to the ad agency to do it, and that's not too close to where you live.
Oh, you have to
So,
go.
It can be done from home.
How old a title do you have to be in the public domain?
Pre-1925. So there's a lot of cool stuff back there.
Absolutely.
In fact, there are a couple of books that they haven't even started. There are many texts that they haven't gotten around to, and of course, this is constantly moving. There's more and more stuff being released, and they do. For plays, they actually, in many cases, do a full cast. So people will record their parts, and then an editor will piece it all together, and you get an audio of the entire play.
Wow.
I wonder if that includes all of Jules Verne.
Yes. Yeah. In fact, they have multiple versions of. So if you want, let me just search up in the catalog here. Jules Verne. They've completed 74 books. They have in multiple languages 20,000 Leagues Under the Sea. And then most of the rest of his works, I think. So Around the World in 80 Days, I'm finding 4 versions. So one of the things about this community is if somebody has already done the book that you want to read, fine, do another version of it. And the flip side of that is if you're listening to one of these and you're like, oh, I don't like this narrator's voice. Just, if it's a popular book, there are probably 2 or 3 other versions of it. You can find somebody else reading it.
Cool.
Great.
Very neat. That's very cool.
So enjoy.
All right. My pick is a website called Stratechery. It is written by one person, Ben Thompson. And what it does, every week there's an article where he analyzes the strategy and business side of technology and media and the impact of technology on society. And they're really long, in-depth articles. And, weekly articles are free. And he does 3 daily updates a week. You have to subscribe for that, but he makes his living doing this. And it's great. I think it's very cool. He's been doing that since 2015. And the articles are really thoughtful. He's got a business bent, of course, and he uses concepts I don't quite get, but I enjoy reading it. He also does a podcast called Stratechery, where he puts a lot of these into words. But I really like the idea that people are still writing long pieces online, and he can make a living of this. And so don't believe it when they say blogging is dead. It's not.
Excellent.
It's very cool. It's very cool. I mean, the idea that you can, this is tech. That's why he can make a living. If I did this for virology, I wouldn't make a living. I'm sure.
Well, this one on top now, the AT&T's decision to buy, in quotes, Warner Media. That's an interesting topic.
Yes.
Yep, it is.
No, he goes into the economics of distribution and supply chains and value propositions, demand. It's just, and he's written a bunch of articles on Amazon and how they work, and it's just great. It's stuff that maybe you don't know about, and he draws little pictures in as well. So really good stuff. He's actually living in Taiwan. He went to the University of Wisconsin-Madison, and She's out there, and it's pretty cool. Anyway, check it out. We have a listener pick from Fernando. Hi, TWiVdom. This Wired article is a great summary of more technical material I shared earlier on the 5 microns controversy. History of science behind it is fascinating. So this is a cool article called The 60-Year-Old Scientific Screw-Up That Helped COVID Kill. It's basically the idea that WHO wouldn't admit that tiny droplets could carry SARS-CoV-2, and so they didn't recommend masks enough and all that stuff. And this is a person who, an aerosol scientist at Virginia Tech, who basically reviewed the history and set everything straight. And both CDC and WHO subsequently revised their recommendations to wear masks and never acknowledged, never made a press conference about it, never really made a splash saying, we're wrong, sorry. But just to be clear that there still is transmission in larger droplets. But these tinier ones can happen. It's just, not as frequent, most likely.
I think Clarissa sent me this.
Oh yeah.
So I'm familiar with it, and as I said at the top of the show, it illuminates the arbitrariness of this 5 micron cutoff, and also I think does a really good job in summarizing the whole controversy.
Yes, it's a good article.
If you're interested in the whole airborne thing, this is a good resource.
And her article that she wrote, this Virginia Tech person, it's linked to. You could get it. It's downloadable. It's in there, embedded somewhere in the text. So that's the original article. All right, that'll do it for TWiV 759. The show notes are at microbe.tv/TWiV. If you want to send a question or comment, TWiV at microbe.tv. And if you like what we do, please consider supporting us. microbe.tv/contribute. Amy Rosenfeld is here at Columbia University. You can find her at antivirus.net. Thank you, Amy.
Thank you.
What'd you say?
Thank you. I had a good time. What? Am I nuts?
No, your audio got cut off a little bit.
Yeah, no. Thank you.
Dixon Despommier is at Racaniello.org and livingriver.com. Thank you, Dixon.
You're welcome, Vincent. And just before I leave, I wanted to give a shout out. I forgot to mention this. Anthony Olszewski, who works for JerseyFreeBooks.com, sent me a book recently on photography of African animals. And he writes, this recently arrived at Jersey City Free Books. I thought you might be interested in, it might be something of interest to you. TWiV long and prosper.
TWiV long and prosper.
Excellent.
So he watches the show, obviously.
Yes,
so
he does.
Thank you so much.
Yeah, Anthony.
I had a great time, by the way. Sorry I was a little bit late.
Long-time listener. No problem, Dixon. I don't know, life calls.
Life does.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com and Alan Dove on Twitter. Thank you, Alan.
Thanks. Always a pleasure.
I'm Vincent Racaniello, and you can find me at virology.blog. And I never say this, but I really enjoy these as well all the time. Chatting is great. We just let it flow, and I think you learn interesting things. So always fun. I want to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.